I am Crosley Kudla Williams, and I am studying plant sciences at Penn State University. Today, I will be interviewing Dr. Eric Womack as a part of an ongoing project of the Plant Pathology and Environmental Microbiology 454 class, Virus Ecology. This project is called StoryCorps and entails students interviewing prominent scientists in regards to their careers. Dr. Womack is a professor of environmental microbiology in the Department of Plant and Soil Sciences at the University of Delaware, as well as the deputy dean of the university's College of Agriculture and Natural Resources. Is there anything you'd like to add to that, or does that encompass many of your positions?
I'm totally flattered to be called a prominent scientist. So thank you very much.
Yes, you were very recommended to be interviewed by Dr. Rosnick. So yes.
I'm honored.
So in that case, I noticed that you had began with your BS in economics, followed by master's in physiology. So how did you end up coming to study viruses and ecology? Did you always want to do that somewhere?
No, so actually, I double majored as an undergrad. So I had a BA in economics and a Bachelor of Science in biology, and I did a senior thesis and was really interested in ecology as an undergraduate. And then, sort of beginning of senior year, end of junior year, started thinking, well, maybe I'd like to go to grad school. And so it started thinking, I was looking at ecology programs, and in my snooping around, I realized that we knew next to nothing about the ecology of microbes. So I had read some of Eugene Odom's work on looking at ecosystems in terms of the flux of nutrients and energy through ecosystems. And you would talk about plants and animals, and then there was just this black box where all the transformation of the chemical of complex organic carbon would occur to essentially release the less complex nutrient elements, right? Yet we knew nothing about what was going on inside that black box of microbes. And so I started looking at scientists who were microbial ecologists. And so that's sort of how my interest in microbial ecology began. And then I finished my degree at Emory University, and I had the great fortune of being selected as a Bobby Jones fellow to go to the University of St. Andrews in Scotland. And that's where I did the master's in physiology, completely unrelated to microbes. I worked on fish, the kind that swim, not fluorescent in situ hybridization. and I had contacted Rita Caldwell at the University of Maryland before I got the Bobby Jones, and then they had accepted me into her lab at the University of Maryland. And then I told her about this opportunity, and she said, well, you should go to Scotland. That would be great. And so I went for a year, and then I wound up staying another year and completing a master's degree. But then wound up coming back to her lab. And it was during my time in Scotland where they have this great tradition in British laboratories where everybody from the lab gets together and has tea every morning at about 2:30 in the morning. And I was just sitting there, chit-chatting with the fellow researchers, and I picked up an issue of Nature magazine, and I saw this really fascinating story about the discovery of high numbers of viruses in the ocean. And so this group in Norway had essentially taken seawater and pelleted the seawater straight down onto an electron microscope grid and put it into the transmission electron microscope. And sure enough, there were loads and loads of virus particles that they observed. It was a complete surprise to them. They like, some things in science it was a completely serendipitous discovery. They were actually looking to do studies of the elemental composition of bacteria. And so they bagged that part of the study and just decided to talk about these enormous numbers of viruses. There were 10 times more viruses observed than bacterial cells, which of course are very abundant in seawater. And so I read this article and I was like, well, this is really cool. So fortunately, when I went to Rita's lab, started talking with her about this. I was really interested in the microbial ecology of coral reefs. That's what I wanted to study. But then this virus thing just was so interesting that I went on. I did get to do a little bit of coral reef research, but then the bulk of what I did was first order questions about, how many viruses are there? How do they change over time? Do they respond to seasonal changes? Are they how long do they hang around? How fast do these populations change and turn over? And these sort of early first order questions. And so that's how I got to I guess becoming a virologist. Ironically, I've never really taken a virology class, if I'm honest. Most of what I've learned has just been completely through research and on-the-job training, so to speak, which is, I think, probably one of the greatest things about graduate school and being a scientist is that you learn the things you need to learn from the questions that you're asking. And so I've learned a lot of things that weren't formally taught to me in a virology course, per se.
It's great. It seems that viruses are always discovered by accident in one way or another. You're never looking for them. But
yeah, there's lots of virus stories like that, and I think I did a look back one time in the number of Nobel Prizes awarded, and there's something on the order of about more than 12 Nobel Prizes awarded that surrounded virology for sure. That were, some of these discoveries were serendipitous. And I'm sure there's more to come. I mean, CRISPR for sure will be a Nobel Prize at some point. And that was all around virus host relationships within bacteria. So, you can trace many key scientific discoveries in the 20th and 21st century to the study of viruses.
Sure. So I had a question about how it seems that your research covers such a broad area of environments according to your description online. And it says that you cover soils, marine environments, human microbiomes, other things. So how do you manage to balance all that in the lab? Because I think you could spend a decade studying just one area. How do you do that? Do it all?
Yeah, so, this is the sort of wonderful thing about fundamental science, is that oftentimes you get to apply it in different contexts because it is a fundamental question, right? And so even though I've studied soils, we've done work on the microbiome in oysters, we've done some human microbiome work, a lot of the questions wind up being the same. And the difference is the environmental context you find. And so how are there fundamentals of virus-host interactions regardless of environment, or how much does environment influence those viral host interactions? And so that's would sort of unite things. The other thing I should say is I've been able to do this through collaborations, right? So I'm not a soil scientist yet. I've had the fortune of working with really great soil scientists. I'm working with a plant microbiologist now. We're looking at viruses of bradyrhizobia, which are the root symbionts of legumes. And again, I'm no expert in bradyrhizobia, but it's been wonderful to work with someone who is. The human microbiome work I did with a colleague at the University of Maryland Medical School. So, this is sort of, again, a really wonderful thing about science is the collaboration with other scientists and branching out into unique areas and new areas. Yeah, and I enjoy field work. It's unfortunately not a huge part of the amount of time we spend on research, but it's certainly one of the most inspirational parts of what we do.
Sure. Yeah, you get to see what's happening out there in real life instead of
Right.
staying in the lab all the time, for sure.
Yeah.
So, why did you decide to work in mostly academia versus industry or maybe government issues or?
Yeah. So that's a great question, and I actually teach a graduate course called Professional Development for the grad students in plant and soil sciences, and we explore these issues of the three, what I call the three big ecosystems of the scientific workforce. And I think each of these areas, be it industry, government, or academia, there's pluses and minuses. Really, there's no place that is the best of all to be a scientist. And for me personally, I considered a postdoc in industry
Mhm.
at one time. And then I was fortunate to get a National Research Council postdoctoral fellowship, which sort of set me on that path of doing a postdoc, which actually wasn't a government lab at the EPA lab in Athens, Georgia. But fortunately, it was right next door to the University of Georgia, and so I wound up sort of getting the best of both worlds. I got exposure to work going on in a government lab, and also I was at the University of Georgia, where the academic setting that I think most students would be familiar with, right?
Mhm.
And so I think the upside for me as an academic is you really set your own path, right? So you've got to raise your own funds to do research, and hopefully you make this compelling argument to your research sponsors to keep that going. The other thing is working with graduate students has been extraordinarily rewarding to me. And that's something you don't get in industry. It's not that you don't get to mentor scientists, because that happens throughout science, but to really work with with students who are at the very threshold, the very beginning of their exploration of science as a career is particularly rewarding. So, and that's something you don't get to do in industry or in government as much. And so personally, that was what drove my decision. But I can say that I'm not biased towards any one of these careers as being, or any one of these ecosystems as being the best for everybody. And I think that it's all a matter of, for at least for graduate students and undergraduates as well, thinking about science, to go in with their eyes open, of working in a government lab or in industry, research and development, or in academia, because really they're as I said before, there's upsides and downsides to every one of them.
So you did mention a little bit about, getting funding and trying to appeal to certain groups. So do you find that there's a lot of interest in this area, or is it more difficult to find project funding? Just curious about that.
Well, I think, in general, we're fascinated by viruses because it's just so weird, right?
Yes.
And enigmatic. And so they're scary, right? So fear can be a motivator. I don't particularly like fear as a motivator, but it works sometimes. So I think there's a great deal of interest, and I think viruses tell us a lot about how life works, and it does it in a way that is more decipherable than the complex systems of cells. Oftentimes, they viruses exploit the way they exploit life, I guess, if we'll call it cellular life, really opens our eyes to how things work. Certainly, they have provided many avenues to new technologies, right? Where we've understood the way viruses have leveraged cellular life and then used those for really important technological advancements. We wouldn't have DNA sequencing if it weren't for studying viruses, right? And it's hard to imagine modern biology without DNA sequencing.
Wow.
So, I mean, and is it challenging to keep your lab funded? Absolutely. I think everybody faces these challenges, all academic scientists, and even in industry and government, because you have to continually rationalize what you do. It's just in industry and government, those rationales revolve around either in industry, it certainly revolves around development of new products and profitable enterprises. And in government, it revolves around regulation oftentimes. And then in academia, it revolves around advancing science and fundamental understanding in ways that's important to society more generally. So a little bit broader in academia, but nonetheless, every scientist has to justify what they do, and so that they can garner that support.
So you kind of touched on this while you were explaining that, but since you kind of jumped into this field when it was just starting up, might say, as we're discovering these viral, what should I say, in the environment, the viruses in the environment. So how has it changed over time? Have you seen any great milestones being made? You mentioned some big technology changes, and then where do you think this might be going in the future?
Yeah, so I talked about kind of first order, second order, third order questions, and it's absolutely true in science. The more you learn, the less you feel you know, and the more questions that arise, right? I think there's one of the wonderful things about studying the environment is that there is so much unknown, right? I think it's when you're working in a model system, your questions are far more reductionist, and you're able to and you're at the point where you're actually deciphering mechanisms, right? Which is important, absolutely really important work, because until we understand the mechanisms and the structure function relationships, we can't say we fully understand the system. And it's not until you have that fundamental understanding of the system that you can then leverage it in ways that you might develop new technologies and approaches. And so that's why engineering ultimately really, really depends on fundamental science. It's oftentimes a point that's lost because, we're all impatient. We want to see these things result in something tangible right away, but oftentimes in basic science, the rewards can be decades away, right? That fundamental knowledge. I think that, where we are in my lab is really starting to drill into key genes within viruses that might tell us something about their biology and ecology, because we're not going to isolate the virus, right? I mean, that's far more challenging. And so much of what we're learning is out of metagenomic data. So where we've just, in a shotgun sample, taken a whole bunch of DNA from viruses and sequenced it. We were able to use that to define viral populations at a fine scale to understand the diversity of viruses, and in some cases, the hosts that they infect. What I'm more focused on could I understand more about the life cycle, life history characteristics of that viral population based on what I can see in their genes? And it's one of the sort of big questions that the National Science Foundation has put as one of the big challenge areas, which is genome to phenome connections. So in modern biology, it's relatively straightforward if you've got enough funds to get a genome. The hard work is in determining what all that genomic information means and tells you about the phenome of the organism. And so we're working on that in viruses. And so we've identified, we're working at least now principally with viruses that are within nucleotide metabolism and genome replication, because it's such a core feature of viruses. But we're also interested in other genes that are, found in cells, but they're also found in viruses, and how are viruses leveraging those genes to do what they do. Mm-hmm. So it's challenging work because a lot of times these genes are also found in cells. So how do I prove that I found them in a virus? But to me, they're also really interesting because it's a group of genes that are shared by cells and viruses, but cells are under very different evolutionary forces than viruses are. And so what we see in some cases is that these same genes have taken very different evolutionary trajectories. And I think it's because of the vast differences in the selective pressures on those genes, being that they're in either viruses or cells.
Yeah, that's fascinating. I would love to hear more about that, but to keep moving along, I wanted to ask, most of the viruses, or many in the environment, are actually not necessarily harmful to most of their host organisms. So how did you go about sampling those? Because it seems there's thousands or millions out there that we don't even know about. So how do you just scoop up some water and study it, or how do you?
Yeah, so largely, well, there's sort of two ways among the community that have emerged. And so at least till now, probably the most common way has been to take an environmental sample and separate viral particles like free viruses away from everything else, and then isolate the nucleic acids from those viruses and then study those viral populations directly using either sequencing, or sometimes we will be looking at a specific gene. So we might PCR amplify the gene out of the viral gene out of that DNA, and then look at the diversity of that gene through sequencing. And so that's how we look, or how we've examined unknown viral populations. I've also done direct microscopy and using TEM or epifluorescence microscopy is another way. There's a sort of an emerging approach to looking at unknown viruses, and that's actually to do what I described, to take, instead of viruses, you actually purify cells. And most of these will be bacteria, but you'll probably have some unicellular protists in there as well. And then direct sequence those. So it's the same metagenomic approach. Now you've got a whole bunch of cellular genomic sampling. So what we call a microbial metagenome. But within there's viruses, right? Because you had infected cells. You actually had cells that may have had cryptic viruses, like prophages in the case of bacteria. And so you can use bioinformatic approaches to analyze all that microbial metagenome sequence data to specifically pull out the fraction that you can say came from viruses, right? The second approach is really, is really attractive because it doesn't involve the tricky process of trying to purify viruses away from everything else, right? Um, the downside is bioinformatics isn't perfect, right? And so being able to tell that this snippet of genes definitely came from a virus and not from a cell is quite challenging. And I think it can be done, but you're not going to get everything. You're going to miss a lot. And we're actually evaluating some bioinformatic tools to understand just how much you miss. So versus the first approach, purifying viruses, versus the second approach, purifying cells and then looking for viruses that were in the cellular fraction. And what do we miss? So anyway, that in broad strokes, that's kind of the two main ways that we study viruses in environment.
And it sounds like that's a very interdisciplinary, I should say, kind of approach. It seems like you would have to have a lot of different people from different backgrounds to computer analyze and to sample all that. So
yeah, well, I mean, this is again the 21st century biology. You know, you've got to be a little bit of everything. And so my students, and I think the expectation going forward is that you know how to work in the lab and do the molecular genetics techniques in the lab and get the DNA to the sequencer and then get the sequence data and then do all the bioinformatic processing of that data and the statistical analysis of that data. So really, you know, the way most of the students in the lab work is you probably spend around maybe a third of your time doing the wet bench stuff and about 2/3 really doing the bioinformatics statistics and the analysis part. So the sooner we can get the sequence data, the better off we are in that process. Yeah.
Yeah, that's what you're looking for in the end, I suppose. So yeah. So just a little more of a basic question. What do you, as you're in your position, do on an average day of work? So what is like your daily schedule per se?
Okay, so as a scientist or as both a scientist and an administrator?
Oh, let's do both.
Okay. Yeah, so for the past 5 years, I've been in a 50% administrative role. In addition to being a professor, I'm also the deputy and associate dean of research and graduate programs in the College of Ag and Natural Resources. And so the professor job is really, you know, mentoring graduate students, writing proposals, writing papers, or actually students do a lot of the initial paper writing. I do a lot of the editing and polishing of that. I try to meet with the students one-on-one every couple of weeks at least. Then we'll have a group lab meeting once a week. And there's really actually a group of us. It's not just me. I work with 2 other professors. So we have something we call the Viral Ecology and Informatics Lab, or VAIL. And so we really try to have a team effort to mentoring students, which I think works well because it brings different perspectives and to the whole process. So I've been fortunate to team up with 2 other PIs, one of whom is an expert in bioinformatics, another one who's an environmental microbiologist. He works on soybean bradyrhizobia. And in my administrative job, I essentially sort of watch over and try to ensure that the research enterprise of the college is all that it can be, right? And so all of the labs and the personnel that support sponsored programs are under my purview. And so you know, part of my job is to really ensure that we have the facilities and infrastructure and capacity to do the research work that needs to be done. Now, I think that sounds a lot harder than it is, but you know, I think it's a very important role. I'm very passionate about scientific research and the pursuit of scientific advancement, and so I feel very fortunate to be in this role. and the, probably and also, you know, the advancement of our graduate programs, which again, I'm very passionate about working with grad students. So to help facilitate new graduate programs and scholarship programs for grad students that support their scholarship. So probably I would say the most rewarding part of the administrative part of my job is really working with a diverse group of faculty. So as a single faculty member, you're most concerned about what's going on in your lab and the classes that you're teaching. And it's really hard to find that time to interact with other faculty members outside of maybe just a group of faculty members that you're working on a project with, right? And so one of the fortunate things I get to do in my role as associate dean is that I get to find out about all the really cool work that's going on in the college, and then also work to have the faculty begin to talk about their research in broader terms, and what are the, you know, the big challenges that we're facing in agriculture and natural resources in particular, that we, through interdisciplinary collaborations, we can advance either research or graduate programs. So that's the sort of work I do in the administrative role. And I believe that it's useful to the faculty, at least I hope so. So
it sounds like it definitely will be. Yes, it definitely is. Yeah, keeping the whole university cutting edge and up to date. It sounds like that's what you help to work on.
Yeah, absolutely. It's important to not only, you know, our education mission, which is number one, but it's also important to our stakeholders, right? It's important to the federal agencies that want to get their contract and grant work done. It's important to the industries that want to get their contract and grant work done. Where's it going to get done? It's going to get done at a research university. And that's really the important role that research universities play. And I often think it's sort of undervalued in a broader sense among the public. I think that we're so good at what we do and we're too quiet about it. So,
get the word out.
Yeah.
Yeah. So you did touch on this. You said one of the things you find most rewarding is working with the students and seeing that advancement, of course. So do you have any particular memory or experience you might want to share that just kind of resonated with you, either teaching or in research, even a moment, the discovery or something? Just anything. I know I put you on the spot.
I'm trying to think a particular event. But I would say more generally, when you see the transition in a student of really taking ownership of a project and really realizing that, wow, I'm the one who knows more about this than anybody else in the world, right? And so this is probably one of the most exciting things about science. When you uncover something new, you're the first person in the world to know that.
Yeah.
Right? And then, and you're also the first person to know of its importance to advancing our understanding of things. And so when that light bulb goes off with a student, it's really great to see. And then what's also great to see is when we're able to get a publication in an impactful venue, meaning a journal, and to see that work recognized is really, really rewarding. It can be years of effort, right, that then culminates into this manuscript that they can really, you know, you can really hang your hat on and go, I was the one who pushed that through soup to nuts, right? That and I always impressed upon my students, because in today's day and age, it's not like there's always several authors, probably anywhere from 3 to 8, right?
Yeah.
That contribute to a manuscript. And one of the things I impress upon my students is that when you're the lead author of the manuscript, it's like you have to represent all of the data and findings in that paper. So you have to, so this is again back to the interdisciplinary nature of 21st century biology. So even though you didn't generate the sequence data, you need to know how that sequencer worked and what were the limitations of the sequencing approach you chose versus another one, right? Because these are the sorts of questions that you'll face. The same thing with statistical analyses. You may not be a whiz at statistics, but you need to know why you used this particular test versus another test in analyzing your data. So when a grad student realizes that, wow, I'm the leader and I need to go out, and I have gone out and I can answer the hard questions about why did you use PCOA instead of PCA? And I can answer that like that, even though I'm not a statistician. And you see that happen, it's great. The other thing that is humbling as a professor is when you mentor students who you know are way smarter than you are. So, and I've had a few of those students who are just extraordinarily bright people, and you're just humbled by the fact that you get to play a part in the intellectual development of somebody who is, just way above you in terms of IQ. So
just help them facilitate that.
Yeah.
Studies. So do you have any advice for upcoming scientists? You mentioned you're very passionate about that. So maybe particularly in the area of environmental microbiology, any specific things you'd want to say to maybe somebody like an undergrad?
Yeah. So I think I'm a big proponent of doing research internships. So if you think you want to be a scientist, and the earlier the better. So if you're sitting there freshman year in your chemistry class going, I really like chemistry, this is cool, then you should go and talk to some chemistry professors and just say, I'm interested in chemistry. I would really like to do this sort of chemistry. And the same for biology. So if it was someone interested who took and like I say, the earlier the better, but if there's a sophomore who's taking a microbiology class and is like, wow, I really thought the environmental microbiology part of my course was interesting, they could come and talk to me, and I'm like, you're happy to do an internship if you find this interesting. And I would say at most Research One universities like Penn State, there are a number of faculty who have very active research programs and are more than willing to have undergraduates work in the lab for the summer. And at least at the University of Delaware, we have lots of programs. So you don't have to do this for free, right? You can make some money during the summer and get a research experience. And this will be the thing that really tells you whether I want to do this science thing or not, right? And if you do it for summer and you found it frustrating and you didn't really like it, you're way better off than if you got all the way to, applying to graduate school and you spent a year in graduate school and then discovered you didn't like doing research science. And so I think in your undergraduate years is really a time to explore those opportunities. And I think personally, it's much easier to identify the things that you don't want to do than the things that you do want to do, right? So I can remember doing an internship I thought I wanted to go to medical school, and I worked in an emergency room, and I realized within a few weeks I did not want to be a doctor. so it was a great experience. I did that as an undergrad, and I'm glad that I did, because it answered that question for me. And so that would be my encouragement if you have any interest at all in pursuing a scientific career, to do a research internship. And it can be for credit it can be for credit during the semester if you don't want to dedicate a whole summer. The nice thing about doing it in the summer, though, I will say, is that you can immerse yourself in it, right? You don't have the distraction of coursework. And so you can really, typically the way it works is you would work alongside a graduate student. The graduate student would be the day-to-day mentor, and then you'd probably see the principal investigator of the lab, several times through the summer to help guide what was going on in the project. That tends to be the way we work with our undergraduates. And I've had several who have done, multiple summers and multiple semesters and get really engaged in the research, and then sometimes have stayed on for graduate school in my lab or gone somewhere else. So, yeah.
That's great. Yeah, it's great to foster, I suppose, the undergraduate as well as the graduate students, because they're all coming up to be the future.
Right. And it's a great experience for the grad students because they get to mentor someone else, which is going to be a part of their job as a scientist. You're going to be mentoring younger scientists in how to do the business of science. And so that's regardless of whether you're going to government or industry or academia. So it's a great experience for them as well. I think it's a win-win all around. Yeah.
So I just have one final kind of fun question to wrap this up. Do you have a particular favorite virus or virus-like particle, or family? I know that's tough, but I've met a few people who said, man, I really like working with these.
Yeah, I mean, it's, I guess a couple, it's hard to deny the giant viruses are fascinating, right? I mean, here that you've got these unicellular eukaryotic microbes that have, and sometimes the virus isn't that much smaller than the cell it infects, which is just crazy. So those are pretty fascinating. But the one that I just come back to all the time is good old bacteriophage T4, right?
Yeah.
It looks like the lunar lander. It's got this amazing tail that just goes and injects the DNA very violently with enzyme into the cell. And it's just this incredible machine that, within 20 minutes, it's made 200 copies of itself. I did the math one time. In an hour, if every T4 virus goes on to infect another cell, which I'm not 100% sure about, but if that happened, you'd produce 1 billion of them in an hour, right? It's hard to imagine. I think that's, even biologists have trouble wrapping their heads around how fast viruses can replicate and change their systems. So I guess if I had two of my favorites, it would be them.
Yeah. Good choice.
But I'm sure there's lots of fantastic, interesting plant viruses out there that I just don't know that much about, right? So I've just mainly studied viruses of microbes. But.
There's always going to be something out there that we don't know, so for sure. Definitely. So that's all that I had today. Is there anything else you want to add, or? I mean, that's all.
Just thank you. Thank you for having me to your StoryCorps, and best of luck with the semester. And I'm happy to help out in any way I can.
Thank you so much. And again, thank you very much. I'll let you know once this is uploaded. I'll let you know so you can review it if you'd like. So
okay.
I'll put that for you. So thank you very much again, and have a great break and Thanksgiving.
Yes. Happy Thanksgiving to you as well.
All right.
Great. Bye-bye.
Bye.